Welcome everyone to the Coastal and Ecology session. I am going to just quickly introduce all of our speakers, and also just a couple of housekeeping things. You can put your questions into the chat. We will be monitoring the chat. We will have each of our speakers present and then take questions from you so that we can track your questions for each speaker. But if you have questions that go to everyone, just make sure that you note that in the chat. Also, if you need to listen in Spanish, we have that available to you, and so that you know at the end of the breakout sessions to get into the larger session, you have to go back to the Zoom link from this morning. So just a couple of logistics things. So my name is Lisamarie Carrubba. I work for National Marine Fisheries Service, and I work in the Office of Protected Resources. And I am here today in part because, as I'm sure many of you know, species such as corals, particularly 7 species of corals in Puerto Rico and Virgin Islands, are listed under the Endangered Species Act largely because of the effects of climate change. So that is my interest for being here. And we have some speakers today who are going to talk with you about our coastal and marine resources and the effects of climate change on those resources. So let me just quickly read everybody's bios. So Dr. William Hernández is the founder and senior consultant of Environmental Mapping Consultants and currently works as a researcher for the University of Puerto Rico, Mayagüez. He has over 18 years of experience developing and implementing GIS and remote sensing applications for a wide range of industries, specializing in environmental and coastal marine ecosystems. William holds a PhD in marine sciences with a specialty in biological oceanography from the University of Puerto Rico and postdoctoral studies in remote sensing and water quality in coastal areas with City College, City University of New York. And his presentation will be satellite and field water quality parameters associated to coral reef areas in Puerto Rico. The next speaker will be Yaritza Rivera. Yaritza is the Marine Debris Hurricane Response Coordinator and oversees and provides support for marine debris related projects, activities, and actions in response to marine debris generated by hurricanes in different regions. She also assists regional partners in developing projects to assess and prioritize objects for removal, complete environmental compliance procedures, document best practices, and ensure appropriate removal and disposal of marine debris. And Yaritza will be talking to us today about NOAA's marine debris program in the Caribbean, prevention and recovery efforts. Next, we have Áurea Rodríguez Santiago. Áurea holds a PhD in biological oceanography from the University of Puerto Rico, Mayagüez, specializing in marine zooplankton ecology with an emphasis on fish larvae. She has worked in the field of science education and research at different levels in Puerto Rico and the US, including mentoring teachers and students K through 12 to university level. She is the founder and director of Taller Ecológico de Puerto Rico, an environmental education, consulting, and community science organization. And the title of her presentation is Zooplankton Biodiversity and Dissolved Oxygen Patterns through Seasonal Sargassum Pulses. And then last but not least, Hector Ruiz earned his doctorate degree in marine biology from the University of Puerto Rico in Mayagüez with an emphasis in marine botany, coral reef organism taxonomy, and ecology. Dr. Ruiz is the executive director of HJR Reefscaping. And through this venue, in collaboration with local and federal agencies, various environmental restoration projects have been completed in mangrove cays, coral reefs, seagrass habitats, and with fisheries resources. And the title of Hector's presentation is Understanding Coastal Vulnerability in Small Cays Around Puerto Rico and US Virgin Islands. So we will go ahead and get started with William. William, you ready to go? You can feel free to turn your camera on if you want, or you can keep it off. I know that Hector and Áurea are going to keep theirs off just because they don't have, they're on generators right now due to a lack of electricity. So William, go ahead. The floor is yours.
Thank you. Thanks, Lee. And thank you for the opportunity to present here. I'm going to leave the camera on just in case, but if I see a little bit of lag, then I'm going to turn it off. And sorry about that. But again, like Lee mentioned, I'm going to talk a little about our current projects related mainly to using satellite observations on products for water quality parameters, specifically associated with high priority coral reef areas in Puerto Rico. And these are some of our co-authors and collaborators, which have helped in different tasks and part of the projects that we have, that we're currently ongoing for this one. Let me see if I can, next slide, sorry about this. So here's an overview of what we're going to try to cover today. And mainly we will focus on how and why we use remote sensing for water quality and the different study areas, and some findings and results that we already completed, and some of the next steps that we're currently working towards for this project specifically. This is a picture of San Cristobal area in La Parguera, those you know that site. And this is one of the sites that we're currently evaluating for our project. And presence there, you almost cannot see it, cervicoids there, but it's a nursery site that is currently ongoing and is under use. So to put things into context, first, as we all know, I mean, land-based sources of pollution are considered a major threat for corals, and not only because the promotion of disease and mortality, but also the disruption of the critical reefs ecological functions. And also, as we're interested in the remote sensing component, we see a lot of light attenuation associated to the reef areas, and the effects of these land-based sources of pollution can augment current stressors, like, for example, rising sea surface temperature that promote coral bleaching. So I get this question a lot, and related to this project, why use remote sensing? Well, the color of the ocean tells us a lot of information, and the pictures on the right that are from Guanica Bay and Oceanic waters, you can see the difference in the color of the water, and that difference is related to water constituents, which are mainly suspended sediments and from turbid waters, color dissolved organic matter, and also the presence of phytoplankton and zooplankton, and the chlorophyll concentration in the water can change the color. So we use satellites mainly because these provide a synoptic view. So we get a screenshot or a snapshot of water quality conditions around a larger area, for example, the whole island of Puerto Rico. And we also use them because they provide us what we call continuous monitoring or virtual stations, which in this case is a pixel. I mean, we select that pixel and collect information available and products available from satellite imagery that has been processed to generate products from these virtual stations. With the benefit is that most of the information that we have can go back 10, 20 years, and it can be used to provide baseline information. So we can then analyze baseline data and trends of those locations that could help us provide a better picture of the locations and areas where coral reefs are located. Some of the products that we use from satellite, and we're not going to go into a lot of detail, but are mainly the chlorophyll A concentration, which can be used as a measure of productivity or nutrient status, the KD490, which is a turbidity value, if we put it that way in simple terms, but is the diffuse attenuation coefficient at 490 nanometers. So that diffuse attenuation takes into account information not only from the chlorophyll concentration, but also from suspended sediments, dissolved organic matter, and other water quality constituents. And the KD par is more or less the same as the KD490, but it's focused mainly on the photosynthetically active radiation, so directly related to photosynthetic organisms. And the sea surface temperature, even though it's not a water quality parameter, but is often we collect information because it is associated with stressors to coral reef areas. And the images on the right, on the top, represents a moderate resolution sensor, in this case the VIR sensor at 750 meters. And this is from a rain event that occurred in 2014 in La Parguera Guanica area, which took about, we got about 6 inches of rain in a 3-day period. And the image on the bottom is a Landsat 8 image, which is a 30-meter spatial resolution. So you get, when you combine these two types of information, then you can get a more complete assessment of the area. the image on top has values that can be used and measured, but the image on the bottom just gives us a false color image that we can see the plume, or we cannot retrieve necessarily data from it. So this is something of the trade-offs of the different imagery that we use to detect information from coral reef sites. But we use, in our project, we were more focused on combining this information into a more meaningful database to be used. So based on these, we then were focused on developing a scientifically sound remote sensing and in situ water quality analysis. First, to establish some baseline water quality data over coral reef areas, mainly in this case, coral reef nursery sites. Not only use satellite remote sensing, but also validate that information for water quality over those areas in hopes of providing management recommendations on water quality and potentially providing some thresholds. So in the end, maybe some standards could be applied or adopted specifically for these parameters for different areas in Puerto Rico. These are some of the sites that we worked out through the project. The green sites, the green points, sorry, are coral nursery sites. The red points are areas identified as priority areas for restoration. And the yellow boxes are areas that have been identified as high turbidity areas of exposure to high turbidity events from previous projects that we've done in the past. And the green box is a NOAA HFA area or habitat focus area in the Northeast. So besides analyzing satellite data, which in this case were analyzed about close to 12,000 images. So we collected daily images from the database. We also collected field observations, not only for water quality, in this case chlorophyll, suspended sediments, and turbidity, but also optical data for KD490 and PAR to validate what the satellite was seeing in space. And also we deployed long-term temperature loggers, and this was done in collaboration with the NOAA Restoration Center located in Aguadilla. So those loggers have been collecting data from the start of the project around 2019 at various locations. And these are some of the results. I just want to, this is an animation showing monthly summaries of KD490. And sorry, the scale didn't show up, it was cut. But the red values, the red colors, sorry, indicate higher values of KD490. And you can see the monthly transition. And when you start getting into the rainy season, you see colors kind of surround the island. And you can even see plumes and waters from the Orinoco coming into the southwest part of the island. So this is some of the products that we can derive from these huge database that we analyzed. If we want, another option that we have is to analyze individual products. In this case, more a regional type approach to understanding the information. And in this case, this is a graph of the KD490 product. The 0.1 value shown here is a 0.1 value is a good reference to show that values above 0.1 are considered areas exposed to turbidity, and values below 0.1 areas considered not exposed to high turbidity areas, high turbidity values. And these are some stations that some sites, sorry, that we collected from different areas. For example, all these sites represent areas in Southwest Puerto Rico, and these areas represent areas in Northeast Puerto Rico. And you can immediately see differences, the variability in the Southwest Puerto Rico sites all around the time series that we had here from 2002 to 2021. And the areas in the Northeast Puerto Rico have been more stable throughout the years. And this is based from satellite mainly, because we only started collecting field information from 2019 only. When we analyze this information on a monthly basis, we start to see then the seasonality in the data, especially in the data from the Southwest Puerto Rico, where you see the rainy season, the first peak here around May, June, and then the larger rain season peak around September, October range. at least for the southwest stations, is very dramatic. It's present in the northeast station, but it's hard kind to define that information precisely. Again, this is the all daily data from all Januaries for the past 20 years. So this is a lot of information inside these values. Another way we can analyze also this information, and we're currently working out to put this into a database online, is individual sites. And the colors correspond to the colors on the graphs. So the Andrea station, which is a little bit more oceanic, you can see the values way below 0.1 and representative of an oceanic station, as you would expect. But when you analyze the Cayo Coral East and West sites, you see that the Cayo Coral West is a higher value, even though it has the same curve. And this is because it's exposed to the Guanica Bay plume coming out of Guanica Bay. So it's, even though it's not so far apart, you can see that difference in the satellite data.
William, one more minute.
Okay. We also compare the in situ data collected from SSTs, and this has been going on from 2019 only. But you can immediately see a rising trend from 2020 to 2021 for these sites. We had some data missing, and it's because of instrument failure and the COVID that limited us the amount of time we were out. But when you expand that to the database for satellite, you can immediately see trends associated with this. And this is a 2005 high temperature event that occurred that resulted in high bleaching for corals around the island. 2010 event, sorry. Again, but the trend here now is rising, and it's rising steadily from 2019 to 2021. And you can see even individual differences between the sites. So this is something to take into account when we're analyzing sites for determining sites for coral nurseries as well.
William, I'm sorry. I'll give you another minute, but wrap it up, please. Thanks.
Okay, yeah, this is the last slide, sorry. All this information will be contained in a web mapping application that I will share the link so you guys can start accessing that. It's still on the draft stages, but most of the information will be presented there. And in summary, well, we were focusing on developing new tools to use the available satellite data and sea surface temperature data, so it can be used for assessing providing management recommendations and establish at least some thresholds for water quality parameters around the island. And our next step is to finalize this information and data dashboards. And we would like to thank the NOAA CRCP program for funding and supporting this, and also the NOAA Restoration Center, Sea Ventures, and HJR Reefscaping for the outstanding field work and support with temperature loggers. Thank you.
So the question for you is, how can we overcome the challenge of data being largely limited to near shore sites?
Yes, and that is a good question. Most of the data that we're using is 750 meters to 1 kilometer data. So it allows us to go a little bit closer to shore. However, we're now, as I mentioned, for sake of time, I didn't include the high resolution sensors that we're analyzing. We're now using the high resolution sensor and applying the ocean color algorithms. So retrieving product from chlorophyll and KD490 for the high resolution data. And some of these sensors can go over every 5 days. So we have pretty good resolution, and the spatial resolution is around 10 meters. So we can go very, very near shore areas. However, it's still a limitation, especially in areas where you have a lot of cloud cover. So it's still a challenge, but we've slowly overcoming this. Specifically for a specific area, we've seen a lot of improvement.
Now we're going to move on to Yaritza. Thank you very much, William. That was really fun to watch those animations, although depressing at the same time, perhaps.
Thank you for the opportunity. I'm just going to be in representation of the Marine Debris Program from NOAA. And as an overview, I'm going to just give a brief presentation on what is the Marine Debris Program per se. And as a guide, the impacts of climate change and coastal and marine resources, strategies to mitigation and impact, and challenges to implementing these strategies, all focusing on marine debris. So the NOAA Marine Debris Program was established in 2006 by Congress as a lead for marine debris efforts in general, with the vision of the global ocean and its coast free from the impacts of marine debris. We currently have 6 goals for the 2021 and 2025 strategic plan, which prevention is the ultimate goal, just to make everybody start preventing the marine debris in first place to enter in the environment. But we also support removal of debris, research, emergency response, which is my line of work mainly, detection and remonitoring, which is a new goal that for this strategic plan, and regional coordination. On this one, we have regional coordinators around the United States, but also Florida in the Caribbean. And what do we do in general? Provide regional expertise, but because we have constant contact with the partners and stakeholders, which we keep coordinating all the time, and facilitating the creation and implementation of action plans, which I'm gonna be talking about that too. But we want to know what's happening in the Caribbean. Your points of contacts is myself, Yaritza Rivera, as Lee mentioned, I'm the Hurricane Response Coordinator, and Ashley Hill, which is supporting. We're basically working together in all projects in the Caribbean and also in Florida and other projects as well. But there's a lot of things going on regarding the marine debris program. We have different removal grants in both USVI and Puerto Rico. One that I'm going to be highlighting is the Hurricane Supplemental Projects. We also have emergency response guides, and the USVI have completed their marine debris action plan. And we are in the process of building it right now. In addition, here in Puerto Rico, I'm based in Puerto Rico, we have an abandoned and derelict vessel inventory happening right now with one of the presenters, which is Hector Ruiz. Impacts of climate change in coastal and marine resources. Why do we have to care about marine debris? I'm pretty sure that all of you remember that 2017 with 2 major hurricanes impacting the Caribbean. Definitely is a year to remember. During that year, there was an emergency support function initiative, which means that the Coast Guard was involved in removing around 500 vessels from the Caribbean, from USVI and Puerto Rico together. Why do we care about vessels? Vessels have shown to be well, terrible if they run aground. They have a huge impact in coral reefs. The DNR is actually really active in emergency related to vessel groundings. And we have had cases of like millionaire costs of impacts on coral reefs, also seagrasses, nesting beaches, etc. So during that hurricanes, we had, as you know, a lot of vessels that just went like run aground and different things. This is an example from USVI, and this one even like takes a long time to remove them. This one even has some cacti growing on them. This one, for example, is from St. Croix, or was, because gladly we were able to remove it. There was even like a hazard, not only for different ecosystem, but like for safety. Seaplane crashed with this one in St. Croix. There's also other targets that we have been identifying. This one is preventing the ecosystem from the recovering. It's in Crown Bay, St. Thomas. These storm surge and the beach erosion, how they make some different coastal constructions to collapse. And different docks and piers also have been damaged and as a result of hurricanes and major hurricanes. So for mitigating these impacts, we have different initiatives. In this case, like we have some hurricane supplemental fundings. Through the program Marine Debris, the USVI was able to get 4.2 million, and the DNER was able to get 3.7 million to actually remove the remaining debris that the Coast Guard wasn't able to remove after their ESF-10 mission. This is still a work in progress, and it's going to be until September 2023. So more debris to come. In Puerto Rico, they're like we're working in base of contractors in collaboration with the DNER, and so far, I don't know if you can see my mouse moving around, but the green areas is just one company that was that has been removing a lot of debris. They start by the East Coast, Culebra and Vieques, and they actually finished those targets and they removed vessels and other medium to large debris. Then they start working on the Hobos Bay area. They moved to Mayagüez. They were able to remove that dock there, the pier, and then they went back and they're still working in Hobos Bay, where they were also able to rescue some corals. In the dark blue areas, another company was able to remove 2 vessels, and now we're targeting Barceloneta area, which has a lot of collapsing structures and concrete structures. These are examples of the pictures of some of the contractors have share with us some of the debris that has been removed. In the first one, a vessel, second one, like different ropes, fishing gear, etc. And this is an example of one of the first report. These numbers are going to change, but it seems that fiberglass, of course, related to vessels is a big deal when we talk about debris in general. In the case of the Virgin Islands, the Department of Planning and Natural Resources is collaborating with us in the removal of different targets. They divided it in 3 priorities, which is the Crum Bay area, which I showed a picture before, post ESF10 vessel removal, and community-based cleanups. I'm going to brief real quick in the Crum Bay and post ESF10 vessel removal projects. So challenge here is really expensive to remove debris of this type. So they kind of struggle a little bit with the funds available, but they're now being able to start the process of removing soon. In the case of the post ESF10 initiative, they use as a target, like a line of target, like removing 12 vessels in St. Thomas, 18 in St. John's, 8 in St. Croix, and happily to report that the mission accomplished in the St. Thomas and St. John area, and they're still working in the St. Croix, but this is an ongoing project, and we're expecting these vessels to be out of the environment in probably this year. In addition to the supplemental grants, we have other prevention strategies, including like competitive grants in prevention, removal, and research. We have great education tools for the teachers, and also even Sea Grant is building a curriculum for 4 to 12 grade students. Marine debris action plans, which are community plans. These are not plans from Marine Debris Program or EPA, but the community plans. The one from the Virgin Islands was built by the community, UVI, University of Virgin Island. And right now we're working with the EPA in building the one for Puerto Rico. So if you're interested, please contact me. And also the emergency response guides. Our main target is responders. USBI one is published since 2020, and we are publishing the one in Puerto Rico probably in the next 2 months. The other news about this one in Puerto Rico is that it's going to be traducida al español, and this is a work in progress.
It's going to be translated
These guides
to
are
Spanish.
amazing for improved preparedness, for response and recovery after different disasters, and help in the coordination. This is a past project, which another presenter of today collaborated to, Áurea. And why I'm still highlighting it, because they prepare videos for the prevention of hurricane debris. So you can actually access their YouTube videos. They have interviews with different people. They just did a great job. We were trying to empower girls in science by teaching them also marine debris related topics. And finally, abandoned vessel inventory in Puerto Rico. This is a current project, so please, if you spot any vessels, let us know. This one is from Quebradilla, so we're not only targeting the ones that have a PR number, as we call it, but also the ones from drug trafficking and people trafficking. Challenges. What challenges do we have to implement in these strategies? The grant process, as I mentioned, they're competitive. It's a lengthy process, highly competitive, and requires a one-to-one match for Puerto Rico, locally, not for the USVI. What happened if there's another 2017? We got this supplemental funding this time, but will that happen again? Will they be available? We definitely have limited waste management infrastructure. And one challenge that one of the our project PIs mentioned is that gas prices are going up, so they can probably remove less debris than they were expecting. And that's it for me. Any questions?
Will any infrastructure funding be available to help the efforts in Puerto Rico and Virgin Islands?
I wish I have an answer for that question. But it's something that we still don't have specific information. We're still waiting for the Congress to give us more like a green light to know what's going to happen with that. But I'm pretty sure that we'll push forward for that, to have some more funding available for the Caribbean in general.
Yeah, and I will just jump in there and say the IIJA, which is I'm going to get it wrong, but it has to do with the Infrastructure and Jobs Act, the new funding. I know that the Marine Debris Program as a whole is getting money for the next 5 years. So as Yaritza said, hopefully some of that will be allocated specifically to the Caribbean. I don't know that all of those allocations are finished yet anyway.
First of all, I want to thank the Committee of Climate Change for their invitation so that we can share our work. It's a work that we deal with CARICOOS and the Department of Marine Sciences. Priscila Molina, Ernesto Otero, and Julio Morel are also participants, and we're going to be talking about biodiversity of zooplankton and the dissolved oxygen patterns through seasonal sargassum pulses. This is a work that is ongoing, and we are going to share with you preliminary results. And now we are starting a different phase during this month. I know that many of you know about sargassum, but I'm going to give you a brief suggestion. It's a brown algae that spends its life on the ocean surface and floats in large masses in the ocean. It's not toxic. It's actually not harmful. It's an important habitat that provides food and refuge for fish, birds, crabs, shrimps, and many other marine organisms. And recently, it's had a bad reputation, but we're going to see why. Usually, and in other decades, we knew that it was concentrated in the Sargasso Sea in the north of the Atlantic, but we are seeing that there's a new Sargasso Sea in this part that is in the south of the Atlantic. Let's see if that's true. What is the relation between the Amazon and the Sargassum in the Caribbean? Around 2010, Brazil started to explore and export more iron from the Amazon. And that meant that they increased deforestation as well. With deforestation, the soil gets exposed, and every time it rains, the runoff washes away nutrients from the soil. And right now, the Amazon River is the leading resource for eutrophication with excessive nutrients into tropical waters. That means that sargassum blooms in rapid proliferation, and we can see how this sargassum plumage looks like. Studies from the University of South Florida compares the nutrients that are in the sea in two different points, 2010 and 2010. They found that there was an increased delivery of nitrate and phosphate to the Central West Atlantic. On the other hand, on the other side of the Atlantic, we have discharges from the Congo River and the sand from the Sahara that are carried by winds and also increase algae growth. So now we also have that we have the Great Atlantic Sargassum Belt, which I am showing right now, that is extending, and it is continuously being formed and entering the Caribbean and the Gulf of Mexico. The Sargassum from outlook from the University of South Florida, and this is from March 31st of 2022. Now it's showing us right now, and you can go into the page if you want, and you'll find the information from USF. We have seen an increase in the Caribbean in 79%. So we are seeing that this sargassum is entering. And we have here that they published these images. We are in 2022, and it looks a little bit until 2019 if we more or less keep going. Maybe this year will not be as bad as in 2021, which was the case in 2018, which was one of the biggest one, largest one in this decade. The CARICOOS system, you'll find this product in their page, and you can see satellite generated imagery that shows the index of algae and sargassum in this area. So we see here the Puerto Rico case. In terms of our study, as I mentioned, we started in 2010, 2011. We started seeing this accumulation, massive accumulation of sargassum in the coastal areas of the Caribbean Sea and tropical Atlantic Ocean. It would go up to the Gulf of Mexico, and this creates a series of harmful situations for live biological resources. So it's a reduction of light, a reduction of oxygen and pH, and an increase in ammonium and hydrogen sulfide concentrations. And what can happen? The cays can create low oxygen conditions in coastal, especially at shallow areas such as shallow coastal mangrove systems. It will affect different species that are important for commercial and non-commercial fisheries. These are nurseries for these species that are so important, and it can affect species directly or indirectly by affecting their sources of food, all of the network. All of these effects of low oxygen and eutrophication are known to have a wide range of effects on the zooplankton. The biodiversity is key to keep our oceans healthy. They have a specific and important role in the ecosystem, act as an intermediate species, transferring in energy from primary to higher trophic levels. So it's important that we study that. The biological studies of effects have been studied in different parts of the Caribbean. However, the effects in the coast of Puerto Rico is not very well known. We have other colleagues like William and all of the other colleagues that are working with this right now with corals and reefs. So now we're working on this. The objectives of our study is on the impacts of hypoxia caused by sargasso accumulations on the biodiversity, zooplankton, ectoplankton, post-larvae, and fish juveniles and inhabit shallow coastal mangrove systems. With approach or how we're going to approach this program is that we have forestation in La Parguera. We measure dissolved oxygen through different methods. Then in a sampling frequency in terms of biodiversity of zooplankton once per month. But now that we have a new stage, we're going to have 6 additional samplings that will be interspersed during the sargassum season. We position DO loggers for a period of 3 days. So it's going to be during the sampling of zooplankton. These are our stations. You can see that they are in these areas where we have mangroves, and it's pretty shallow. Isla Magueyes, Varadero, Moncio Jose Uno. This is something that is very impacted. And we see it in bold because the results that we're going to present today is from this area that we call Moncio Jose Uno. And we also have a station that is a control station, which is in Cayo Enrique, which is an area that is not as impacted. Where our zooplankton is done by light trap. It uses a chemical light. It's deployed at dusk. It's retrieved at dawn. So it's left the whole night. And the mechanism is that we have some slits here where we have the organisms that are attracted by light, and then they are trapped in a net of a micron mesh that it's in the lower part of the device. This floats. We put it with a block, we set it down, and this is how it looks when it's already in the water. Then we collect the organisms in this pan. We store it in ethanol until we go onto the next spots. Then we also do a plankton net with the things that we have here described. Once we get to the laboratory, we have larval and juvenile fish sorted out. We have the organisms identified morphologically to the lowest taxonomic level possible. And we also take photos and document them. The next step is that we are doing DNA metabarcoding. In this case, a comparison between single species zooplankton. You just have a PCR amplification, you have sequencing, then we have the results for species A, B, C, or D. For instruments that we use for dissolved oxygen, you can see them on screen. We use a polyrographic DO instrument. We also use an HEO-BO dissolved oxygen data loggers, and we also use light and pressure. We put them in this platform, in this pedestal with a weight, and inside is the oxygen sensor. So when we take out the sensor after 3 days, we put them in 3 days, and once we have the net out, we take that out 3 days after. And the results, morphologically speaking, is that we've seen an increase in May when the Sargassum was reaching our area massively. And then in June and July, we saw a increase. And if we look at the results of oxygen, we have a peak in May. And then in June and July, we see a drop, an immense decrease. We're still analyzing this. It might be that because this sargassum probably is bringing in organisms and producing oxygen. So we have the results from the sensors and the YSI that we started in October, but that will be for the next step where we're going to be comparing. We've seen fish larvae. We have seen different types of anchovy. We've seen the sargassum fish, post-larval gray snapper. We've seen crabs, a lot of different types of crabs. And we have seen here the DNA metabarcoding. Even though if you don't understand it, we can see specifically what we have in terms of more detail for the general group. So we have detected a decrease in the zooplankton diversity and abundance associated with low DO for June and July. We have to study that and see how it's part of the normal life cycle. We have to plan long-term monitoring that the DNA metabarcoding is effective for that. And we have to see how this could be important for fisheries and for fishing efforts. It could be direct or indirect, as we see with a yellowtail snapper. So we want to thank Patricia Chardon from CARICOOS, Luis Rodríguez, Eric Garcia, Adolfo González in the field and the office, Maribel Aneda. If you have any questions, I am available.
Thanks, Áurea.
So thank you, Lisa, again for inviting me to present today. This is like a small part of a bigger project that started in 2017 with some funds from U.S. Fish and Wildlife Service here, the office here in Cabo Rojo, and multiple collaborators, National Conservancy, NOAA, DNR. And it was like trying to understand the vulnerability of the small keys around Puerto Rico and U.S. Virgin Islands. If you can go to the next one, please.
Yeah, sorry,
So,
PDF, it's coming
okay,
out
that's
there.
right. So as we know, Puerto Rico and the main USVI islands are archipelagos. They're surrounded by many small keys. And these keys are really dynamic. And anything that happens, like climate change-wise, it's gonna affect and gonna impact these keys first. So we're gonna have like it's gonna show the results of those impact right away, some of them will take some time, some years to, so we can see the effects and the impacts of these like climate change events. But what I'm trying to tell you is like the first messages of how things would change in the coastlines will be seen here in these little keys. They're impacted constantly by rain, tidal flows, winds, and current flows. And lately in the south of Puerto Rico with the seismic activities by earthquakes. So another more stress to these little systems. So I think that I was gonna say one more thing. These like are basically wetlands environment, and they serve like really essential ecosystem services like flood and erosion control, and provide shelter and food for many different animals. You can go to the next one. And to study them, we need like a really high resolution and temporal and spatial temporal resolutions to study them in the right way. So some of the objectives on this project, on the big project, was to really generate a detailed benthic map and terrestrial map of these like little keys to further to assess the vulnerability of these keys around Puerto Rico and the U.S. Virgin Islands. We use like drums for that, and so we actually wanted to adjust the protocols to get the highest resolution images, higher orthomosaic images, so we can actually interpret that and get the information from the habitat. And finally, analyze these maps and this information on habitat to see the vulnerability of them and how climate change can impact them in the future. You can go to the next one. So we started with like these like 16 cases around Puerto Rico and USVI. They were selected actually with a consensus of like many scientists and personnel from the different agencies involved in the project. Some of the things that we consider was the size of the keys or the case for the drums in the drum perspective. If they have like any protected species associated with them, human utilization, and any partners for the future protection and conservation. And among other criteria were used to select these like 16 keys or cases around Puerto Rico and USVI. There was like 12 around the Puerto Rico main island and 4 in the USVI. You can go to the next one. So we developed, before we went to the keys, we developed a hierarchical habitat classification, and really simplified one based on the classification of wetlands and deep water habitat in the US that was developed by Cowardy et al. In 1979. And this is an example of what we came out with. The classification for emergent vegetation mostly, and sandy areas and stuff, and the submerged areas with like corals, like seagrasses, microalgae, etc., etc. You can go to the next one, please. So what were some of the results? On average, most of these like cases, the biggest, the one that had the higher coverage was the coral platform. And there's like, this was like almost expected because this is the baseline of the base of the whole keys. And most of these keys, especially the ones in the south of Puerto Rico, are mostly composed of dead coral skeleton that actually made up the emergent part of the keys. It was followed by seagrass, then seagrass areas in the back area, the protected area of these keys. And in the emergent part, there was like mostly mixed mangroves, black mangrove, white mangrove, and red mangroves, and low-lying vegetation covering most of these keys. There were like 16, on average, 16 different habitats per key. There was no clear indication on the relationship between the cay size and the habitat richness that we can see. And mostly on average, there was like 54 species present per cay. And there was like mostly 50-50, 28 on the emergent part of it, like the vegetations and all the animals in the terrestrial part of it, and 26 on those submerged areas. But it was really important, and something that came out, that the shore habitat on these cays are really important nesting areas for like seabirds, landbirds, and sea turtles. Even the parts with the big boulders, the seabirds use them as a nesting area, and the areas with the sandy beaches are used by sea turtles mostly. You can go to the next one, please. So when we went out to the field, what we did? We just visit the case, took the drum, send the drum up. It will turn out to this like really high resolution image that you see in the left area of this slide. It will come out with a really high resolution. It's like would be like 2.2 centimeters per pixel. You can zoom in and you can even see the tracks on the beach where the sea turtles are nesting. You can see some big, in some of the keys, you can see actually the pelican nest all over the areas. So it was a really high resolution image that you can get out of it. And then we went out too and did some ground truthing stations all around the little islands. And then all this, we gave it to the GIS expert that we have, and she was able to make a really high resolution classification on different habitats on the different cays, like you see in the right portion of the slide. And this is like, this serves us as a baseline of what we had when we started the project. You can go to the next one, please. So this is an example of the images that you can get. And maybe locally, this is Maria Langa in the south of Puerto Rico. We got the images before Maria, the big hurricane category 5 Maria actually hit the island. So in 2016, you can see the whole island with the internal lagoon, all the healthy vegetation, and the two beaches, one in the southeast of the area, and then one in the north. And then in 2017, you can see how actually the center part of the island suffered a big mortality that was mostly like composed of black mangrove in that center area of the island. Hurricane Maria actually mostly like killed all the black mangrove in the center of the island, which was used and is used by many birds, especially the pollo de mangle, as we call it. They use the prop fruits to do the nesting. So there was like the loss of nesting for that bird was like significantly. Even though we don't see it here, but Hurricane Maria too actually impacted directly the coral reef around these islands and keys, actually dislodging many of these like important barriers for this island, many of the coral species, acroporas, orbicelas, diplorias. And that year in Puerto Rico, actually, we were able to secure back or like rescue like 20,000 colonies around the island of Puerto Rico, but there were like many thousands of colonies that were affected and doesn't have the capability to cement back to the seafloor, and that would be, you know, a loss for the island. Then in 2019, by the end of 2019, the beginning of 2020, there were like some seismic activity in the south of Puerto Rico and actually affected this island and actually sink the island 25 to 30 centimeters into the ocean floor. So actually, it actually increases the inundation in the island, it increased the erosion around the island, and actually increased the redistribution of sand in the area. Actually terminated to wipe out all the black mangrove in the island. Then it started affecting the the red mangrove. And you can see in the image to the bottom to the right in 2021 how different it looks from the 2016 image that I showed you. The erosion is like really significant. You can even see sargassum there in the north area of the island. If you can go to the next one, please. So some other keys and some other effects of like big impacts, like a Category 5 hurricane, you can see them in the vegetation. This is like the red area on the image is like the live vegetation in 2016 in Marialanga. And then you see in 2017, after the hurricane, all the loss in vegetation. This is ongoing analysis still, but it's important too to point out that even though in Turrumote, the one island to the top of it, you don't see that much change. But in those islands that don't have that many areas covered by vegetation, a small loss in vegetation is actually more significant and important than in some of the bigger islands that have like a bigger buffer to survive this like catastrophic or like really hard event like Hurricane Maria Category 5 is. You can go to the next one, please. So in some of the keys to the effects on Hurricane Maria, you can see it here. This is like a DEM, a digital elevation model from the drones, the orthomosaic. And you can see how there was a significant reduction in the cover and altitude of the mangrove trees around the island. So that's another thing, you know, maybe it's gonna kill some of the bigger trees. It's killing the bigger trees, the trees with the highest altitudes. So that's another thing to consider as we see how this catastrophic event or really high climate change event changes the morphology and the topology of these like cays around Puerto Rico. And the next one, I think it is the acknowledgement. Thank you for my fieldwork team, Jose, Osvaldo, Luis, Chelsea, Yahaya, Evan, Antonio and Glauco, and the technical assessment group, Nilda, Angel Diepa, Aixa, Lisamarie, and Brett Murray. Thank you for your time, and I'm here for any questions. And I got my power back, so.
So hang on, everybody. I'm going to go back to William for a minute because there was a question that came in for William. So William, is it possible to predict the acreage of coral reefs that could be damaged by a known soil runoff volume? You're still there, right, William? I see.
Yes, yes, sorry, I wasn't.
Okay.
Yes, the short answer is that yes, we can measure, not necessarily predict, but more have a measurement of exposure of that coral acreage or coral site. Saying, for example, we can see if a coral location is exposed by for a certain amount of time to, let's say, high turbid waters or high chlorophyll concentration waters. That is something that we can do with the database that we have spanning 20 years back. So short answer is yes. It will be depending how close to shore it is, and the areas around the island where we get a lot of clouds in the northeast, especially in the mornings, and in the southwest in the afternoons. So those areas, especially in the afternoons, might be, if it is too close to shore, might be a little bit tricky, but yes, we can have that.
Great, thank you. I don't see any more questions for any of our presenters. I'll give it one more second.
I see a question there about the emergent, somebody asked me about the emergent vegetation and what was the...
Okay, yeah, go for it. I don't see it here. Go ahead.
Yeah, actually the person that did the analysis, the GIA expert, he was looking at the reflecting light, the red light. And based on that, she was able to calculate and give us like the image that we have. But I asked for the data. It's still like she's still ongoing and processing the whole data. So I don't have a number of unit of how many area of vegetation was lost. But we're getting into that. Thank you for the question.
Okay, great. I don't see, do we have any?
Yeah,
one more?
yeah,
Oh,
yeah. tenía una pregunta en el chat que me la hicieron directamente. Decía que si el sargazo en fin era bueno para el...
I had a question in the chat. They wanted to know if the sargassum was good for zooplankton. Yes, it is good when it's alive and it brings oxygen and it can bring zooplankton when it arrives. But when it decomposes, now it's not good because there's no oxygen anymore. I answered the person that...
Okay, thanks, Áurea. Thank you all, and buenas tardes, and enjoy the rest of the conference.
Good afternoon.
Thanks.